Okay, this is Akram Vasighizaker. I'm a PhD student in computer science from the University of Windsor. I'm here to present prediction of human pancreas cell types via convolutional neural network and two-dimensional mapping of single-cell data. This is a joint work with my supervisor, Professor Luis Reda and Li Shu. Cell type identification and finding marker genes related to specific cells are of main problems in single-cell experiments. In this work, we focused on identify organizations of cells using self-organizing maps and automatic annotation of clusters using convolutional neural network. One way of cell identity variation is by hypothesizing that different functional processes may lead to different variation in gene expression space. Cell division and mutated cells are of good examples to explain these concepts. There is a common goal in single-cell experiments, that is cell type clustering, trying to present the best group of cells using quantification of transcriptional differences and by calculating cell-by-cell distance metrics after some quality control and preprocessing steps. And in a downstream analysis, there is a need to cluster annotation by cell types using some marker genes. Okay, let us now describe what the main method is. Our method is comprising three main steps. First is to construct gene similarity network via self-organizing map. And the second step, we create images per cell using the template created in the first step. And finally, is taking images as input for CNN to classify them into cell types. Okay, let me describe a little bit more step by step. So we start from gene expression count metrics, and before performing the proposed method, we did some preprocessing, including quality control normalization, just to make sure that we remove irrelevant genes, and also feature selection. In a feature selection step, we use correlation-based feature selection method to select some highly variable genes or marker genes. And this is a list of 13 selected genes after feature selection step. All right, let me describe a little bit more step by step. In the first step, we start from gene expression. We create template by self-organizing map. We can see how genes from higher dimensions are arranged onto a map in which the similarity of the genes are reflected into this map. We can see also the genes that share some functionality are placed close together. And this is an example drawn from our dataset, our experiments. And we trained a self-organizing map using 13 selected genes. If I want to talk a little bit about self-organizing map, these are mainly used for visualization of the intrinsic relationship among data points. Neuron on the two-dimensional output layer or feature map initialized with random weights, and the samples is mapped to the most similar nodes on this output layer. And the output layer is just a lattice indicating position of the input neurons, and similar points are placed in the same group in the resulting graph. In the second step, we create one image per sample by coloring template. So if we have, for example, 40 samples, we will create 40 images. So how we do that? Taking the template, we draw a circle with arbitrary radius around the genes that is placed in the map. And for coloring, we follow the coloring scheme, standard coloring scheme of images in which there are three color channels, including red, green, and blue. And in which channel it is represented via 256 distinct values. And in this work, we use gene expression values as color channel to color templates. Now, once we have constructed images for each of the cells, we pass them through CNN, and then to do the classification, we just use parameters including 32 filters of 3 by 3 And we use a relo activation function, max pooling for pooling, and local response normalization. All right, we have done some experimental results on the Barron dataset, which is a benchmark single-cell dataset of pancreas tissue, including 14 distinct cell types and around 8,000 cells and around 20,000 genes. And then for our data set, we can see a trend in the number of epochs, how the absolute error does on the training set will drop to go to 0 after a few iteration and epochs. And the performance metrics show 97% precision and 96% precision recall. Also, we have done some biological validation. We have taken a few genes and we have seen that those are sufficient for classification. Investigating MCDB highlighted 2 pancreas-related gene sets, including our selected genes 6 and 5 genes respectively, and it shows that those share similar functionality with pancreas cell types, and it's highlighted a strong prediction of pancreas cell types. To conclude the presentation with some highlights, our method demonstrates that we can not only use this scheme for classification, but it's also used for dimension reduction and feature selection. At the same time, also we can use only 30 marker genes to achieve accuracy rate of 98%. And for the future extension, we can integrate multi-omics data in the input layer. I would like to acknowledge the funding agency like University of Windsor, NSERC, and MyTex. And thank you for the invitation for the talk. And if there's any question, I would be happy to answer.